Good morning, ladies and gentlemen. My name's George Morris, and I'm going to be presenting for the next 15 to 20 minutes on ValiRx plc. Our lawyers have had their fair whack at it. I don't expect anyone to read it, but it's there for good measure. What is ValiRx? Well, quite simply, we are a straight, old-fashioned, but highly focused biotech company which focuses entirely on cancer therapeutics with companion diagnosis to go with those therapeutics aimed at specific subsets of patients so that they are treated efficiently, effectively, and with minimal disruption to their lives in what is a very devastating condition for anyone who suffers or those who are around with them. What are we in more detail? Well, quite simply, we have a portfolio of a number of technologies and products which are unique to us. We always hold, except in one particular instance, 100% of the intellectual property rights, and we control that most jealously and with great care. But to make a therapeutic work, you need to know who it works for, how it works, and when it is not appropriate to use it, as much as to when it is appropriate to use it. So we have a therapeutic biomarkers section to us, which basically outlines what a patient looks like and whether the particular therapy suits them, whether it's working for them, and what the outcome of that therapy would be, or what other therapeutics they would require alongside. Our two clinical products are moving through the clinical phase at the rate that these things are allowed to do, which is actually quite rapid, but a great deal slower than I, who manages the process, would delightedly have to say. However, safety and care of patients is always primary. Where have we got to in the last few years? Well, our first trial is a dose escalation study, which is the first in man, through to where we take a compound up to the level where we expect it to be therapeutically useful from all the work of several years that went into it prior to it going into a human being. In this case, since it's for prostate cancer, I can actually say a first in man study without offending anyone too greatly. It is particularly for people with advanced and hormone recalcitrant or hormone refractory cancer, depending on where you were brought up. This is prostate cancer where the treatment is normally to give you the female contraceptive pill, which in males is not a particularly nice thing to do, but it is continuing very nicely, and I will talk about it in a little more detail. We have already shown, and this is the phase 1 part of the trial, that it is safe to any level where we require it, and it is tolerated very well with no serious side effects seen whatsoever related to the treatment. Unfortunately, some of the people on the trial are pretty ill, and they do have serious consequences from the disease, but not from the drug. Also, so far through the trial, we have seen nothing that we did not expect to see from all our preclinical monitoring and the way in which the drug has been used in various models and animal systems. Second product is just about to start in its clinical trial as not a first in man, in that it is not a new drug in many ways. It is a drug that has been used for the last 20-odd years for psychotic indications, and from a rather clever screening technology, it was discovered that in many cancers of a particular type and variety, it showed good suppressive factors related to it. And this has been developed, and it's now called VAL401. I get them confused myself. And we are about to start dosing humans who are in terminal stages of non-small lung cancer, and we expect to have that trial completed within a year, and before the year end, we would hope to be seeing some significant results. In addition to that, we have quite a serious development program on the companion diagnostics and the prognostic diagnosis of our cancers, and this is particularly related to our own therapeutics and the ones we're developing, but also they are useful in other places, and there is a small service business based in Finland where we undertake some of that work. It is profitable, but not a significant part of the business. We also have a very novel, and in fact where the company started from, gene silencing technology, which has been applied to cancer. We have yet to get that into the clinic, in that it is very novel and has a lot of ethical concerns surrounding it, which have been resolved, but the amount of preclinical trialing and testing we have to do has been prohibitive so far. We expect that to progress to a clinical situation within the next 18 months. It is presently being developed with partners in Finland and in Germany, and is showing real promise in various cancers, in particular in breast cancer and pancreatic cancer, but we are some way off putting it into human beings simply because of the complexity of the product. Right, what is VAL201? Well, it's a very novel product in many respects, in that it is a peptide, which for those of you who remember their school chemistry are the building blocks of proteins. It is 10 residues long, or 10 amino acids long. It is a unique sequence, and it has very unique properties, and it is incredibly stable in the stomach solution, which is where we wish to take it in the future. It is also a very novel, I hate using the word very several times over, but I think it's perfectly fair here, where it stops the cancer very specifically, not by interfering with the androgen, which is testosterone, which causes prostate cancer in many people, and it doesn't interfere with that as many of the drugs that do, with all the side effects like osteoporosis, breast growth, mental retardation, respiratory problems, etc., etc. It purely and actively stops the cells dividing, and no other effect from testosterone is prevented or inhibited, which, as a male myself of a certain age, is a considerable comfort. I really don't wish to get a larger breast tissue than I have already, and I would quite like not to be feminized and end up with osteoporosis, which is the standard side effect from most of the therapies. And unfortunately, as we've seen, quite often mental retardation growing. It only, as I said, reduces the tumor's growth, and by so doing allows the body to control this tumor and prevent the overexpression of prostate cancer cells, making the prostate large and proliferating into other tissues. We have follow-on indications with it. Surprisingly, to those who perhaps aren't totally familiar with this rather obscure piece of biochemistry, it's also very good at stopping a very unpleasant female disease called endometriosis, which is effectively like having a perpetual menstrual cycle. If you have a perpetual menstrual cycle, not only do you have the pain and the discomfort associated with that, but you are also probably infertile and are in a rather poor state of mental health as well. That's not to say that periods cause trouble. They don't in themselves, and that I really must add. We are in the process of designing a trial around that specific indication, which we hope to submit sometime towards the end of this year for approval by the regulatory authorities. It is an early stage of design, and we have yet to finalize where and how we will undertake that trial. Nice little graph at the top, which shows a preclinical model of how it works. And you'll see two lines that go up at approximately 45 degrees across. That is the cell's growth and division without treatment. One is just a regulatory control, if you like. The other is a drug that is not 201, but is very similar in structure. And the line marked S1, the red line, shows suppression for the length of the experiment in that particular model system, which was human prostate cancer grown specifically in mice prostates. Difficult experiment to do, they tell me. I'm glad I didn't have to do it. It's a large market as well, and has significant unmet needs. It's not very well treated, particularly in the later stages, or in the very early stages where the treatment is wait and see as much as anything else. Very surprising effect, which came up with the experiment we were doing right at the end, just as we were submitting to put this into first in humans. I had an experiment planned and paid for with our partners, and we suddenly discovered we had the data already and we didn't need to do one extra experiment. So I was left with rather a large number of animals, rather a large amount of compound, and rather than to do what you have to do in such situations of unfortunately putting the animals down, I thought we would use them for an experiment. So we produced these mice with cancer. We injected them again with VAL-201, and we ran them to see whether they would produce secondary cancers or metastatic cancers, in particular bone metastases, which is very common in prostate cancer and is extremely painful, so they tell me. And looking at some of the anonymized clinical notes, it clearly is a very unpleasant situation. And surprise, surprise, and I really was surprised by this, we have not only shown that it did cut the cancer growth again, but it also suppressed the metastatic cancers in a very significant proportion of the animals used. So we know that in very close mimics of the human situation, it does suppress metastatic growth. The trial so far is far too short term, so we're unlikely to see anything in this initial trial in that particular respect, but some of the other markers associated with it are already showing proper inline efficacy. Right, now move on to our second product, which is about to go into human beings as an anti-cancer product. It is a reformulation, fully patent protected, fully covered by intellectual property and all the preclinical work, exactly the same as 201. The only thing we didn't have to do was a lot of very expensive and time-consuming toxicology, because with 25 years human use on this, you can take that as read. And there is a very small trial we did, or a limited trial we did, just to show that the new formulation was as safe as the original. It is. Again, a graph that shows that it works, and that was on a triple dose from a set of xenographic mice again. This is mice where cancer has been grown on them. It really is very unpleasant stuff, but unfortunately you do end up having to use animals in this business. And we are going to be starting this trial before the end of this first half year. It'll be the last 2 to 3 weeks in June. We assume everything else has been done. The drug has been produced in sufficient amounts. The regulatory submission has been made. Preliminary indications is that we will have full clearance to do this. And again, the regulators have come back saying no particular problems, but there is, of course, as always in this business, considerable process to undertake, and we aren't quite there yet. The last one I mentioned was our gene silencing technology, which is quite unique, and we have had considerable success in it, and it works quite simply by finding a gene that is expressing or is working inappropriately in the wrong place. In other words, a rebellious gene. Now, a lot of cancers are caused by this. All the ones that are created by environmental conditions are due to changes in what they call the epigenetics, i.e. genes being expressed where they shouldn't. This gene ice, or 101 product, quite simply finds that gene, sits on it and stops it expressing and doing the damage it's doing. In fact, turns the cell back to normal, non-cancerous condition. We've got a range of different versions of this, one for Alzheimer's, several different cancers, and a couple of inherited metabolic diseases as well, where it has been shown to work in model systems very well. And on the right, again, the classic graph, and as you can see, the light blue line, where a single injection or single dosing has been shown to suppress the cancer growth for, it's about 4.5 weeks, and that's on a single dose. After that, the cells which were not treated begin to become dominant, and of course the cells grow away again. That would be expected, but with a second dose, you would not expect to see that happening. Thank you. Biomarkers, again, they are purely done by us in support of our core technologies and our core products. However, they do have third-party uses, and we do a certain amount of work there. We also have a very good proprietary method of measuring genes, which is considerably cheaper and very significantly faster than the competing technologies without much loss of sensitivity or resolution. In fact, just as good as sensitivity. We just look at slightly fewer genes, but more than sufficient. This is also sold to third parties and is used in a lot of other companies' toxicological screening. There are requirements by the FDA and just coming up with EMA that they do gene screening for specific toxic effects. Where do we sit in the value chain? Obviously, we're not making revenues. We will make revenues, but they'll be extraordinary revenues. We will sell, license, or partner products through. We've had a certain number of discussions, and we're in active discussions with several, exactly when we sell those products on and for exactly what indications. Depends a little bit on the results of the trial and also who comes up with a sensible offer. Academia produces our material. They always think they can develop it. They're not actually very good at developing drugs, but they're very good at coming up with the ideas. Biotech companies fill that gap to the people who are excellent at marketing and selling such drugs, but not these days very good at making them. And they are the large pharmaceutical and the mid-sized pharmaceutical that are effectively marketing and sales organizations. And we sit very carefully in that middle gap of preclinical to mid-clinical work. We then license on to the people who can do the big-scale trials costing hundreds of millions. We would never attempt to do those. We're not set up with 15 people to undertake a 20-center trial in 5 countries with 1,000 patients. It's just not practical. Our partners in this are good. Technologies come from places like Cancer Research UK, Imperial College, clinical trials being done at University College here in London, with a couple of other centres following on, again, well-known teaching hospitals in this country. We work with the German equivalent of DKFZ, which is state-owned and state-run, but acts very much like Cancer Research UK or the Wellcome Trust do in this country. We've been supported heavily by the British government and the European Union for our development of the 201. We're on the second grant, which is completing on that, and we should be, as I said, at the end of that, just about ready for clinic. We also work with an institute in Marseille that's well known for its peptide chemistry, and more importantly, is named after the guy who invented the BCG vaccination, which most of us in this room will have had at school. The biomarkers end is done in Helsinki and in the north of Finland by the Arctic Circle. And we have a JV, and this is one which I was saying earlier, we only own a proportion of, Val401, with a company called Tangent Limited and Seek here in London. Summary, I'm going to cut that short. I've talked enough and nobody's going to have time because I want to go on to the last slide, what's next? It's not particularly forward-looking, so I'm safe there in that all these statements have been made, but it's worth summarising them. We expect to complete the phase 1 and phase 2 trial of Val201 this year. The primary endpoints are safety and tolerability. However, because this has been done in people who have the disease. We are also looking for effects of the drug, or efficacy as people shorten it to. Not quite sure what they mean by that sometimes, but we are already seeing indications that the compound is not just safe, it is actually reducing the increase in cancer. I can't say it's stopping cancer yet. It's too early in the trial for that, but it will do. That I'm assured of. We already are at the pace where we have the maximum applicable dose. We've never managed to give animals or humans enough to show a real toxic effect. Probably isn't possible with this compound, which is extremely exciting. And we are in the process of dealing with the endometriosis trial. At the moment, we still have some issues about how we're actually going to conduct that trial, and it's more of a discussion than an issue, and we will be submitting towards the end of this year for that. VAL401, as I've said already, I spent the last 2 weeks sitting on aeroplanes going halfway around the world, actually, looking at various test sites for this. We've now agreed who the test sites and the principal investigators are, and we are about to start the trial on that sometime mid to late June. 101, we are partnering, and we are going to start doing the formal toxicology again towards the end of the year or the early part of next year, with a view to submitting for clinical use sometime in 2017. Biomarkers are continuing, particularly to cover the endometriosis trial and that area, and we are in discussions with another company who has a different way of approaching endometriosis for a slightly different set of conditions, to work with them to come up with that panel. And we are, of course, discussing seriously licensing opportunities across the portfolio, and would expect to have significant advances on that as soon as the phase 2 part of the 201 trial is done, and when we have in-human data for 401. Board of directors, mixture of business with some science. Despite what people say, there are effectively only 2 scientists on the board. The rest are mainly professionals within the biotech and medical devices industry, or long-term serial financial people. Thanks very much for your time. Our contact details are there, and there is a team of people wanting to talk to you down on stand 82 downstairs. Good morning, and thank you for your attention.